These are the lecture notes for Chapter 12 on molecular biology, which is basically the chapter that covers DNA and the structure of DNA and how DNA replicates and how DNA codes for a protein or codes for our traits. And I apologize because I'm late posting these. One of your classmates let me know that I hadn't posted them, and so I'm going ahead and doing this on a Friday night, kind of late, but that's all right. So I'll go ahead and start. There are 5 sections. The genetic material, section 1, is basically about the discovery that DNA is the genetic material. In other words, the discovery that DNA is the molecule that that codes for our traits. You know, it's not some other molecule, it's actually DNA. So the experiments that led to that discovery, that's covered in section 12.1. And then in section 12.2, the process where DNA replicates or copies itself, which we have already learned that this occurs during interphase, during the S stage of interphase, before the cell actually divides by mitosis or meiosis. So this is going to explain exactly how that occurs. And then section 3 tells a little bit about RNA, because RNA is the other nucleic acid that's involved in synthesizing proteins. And then transcription and translation are the processes where DNA codes for a specific protein. So we're going to go through a lot of chemistry, a lot of specifically how, these different processes happen. So starting with section 1, the first scientist that we're going to talk about is Frederick Griffith. And Frederick Griffith kind of discovered, he wasn't really trying to find out what the genetic material is, but he was studying Streptococcus pneumonia, which is a bacterium that causes strep throat, it actually causes pneumonia, but it's related to the bacterium that causes strep throat. The one that causes strep throat is called Streptococcus pyogenes. But anyway, this one, Streptococcus pneumonia, causes a bacterial form of pneumonia. And he, in his experiments, he discovered something called transformation. And transformation has been really, really helpful in recombinant DNA technology or biotechnology. That process is used whenever you introduce DNA from one organism into another one. So what he did was discover the process of transformation. He discovered that if you put 2 bacterial strains in a test tube together, and one of them is dead and the other one is alive, that the DNA from the dead strain can enter into the live strain, and the live bacteria will now express the genes from the dead bacteria, and that is called transformation. And there was more research by Oswald Avery and some others that confirmed Frederick Griffith's research, that DNA is actually the substance that is the transforming substance. It is the substance that leaves the dead cells and enters into the living cells. So we know that DNA is the genetic material. Before we knew that, when the researchers were kind of looking into what molecule could contain our genetic code, it was kind of narrowed down to DNA or protein. And that is because DNA is a long polymer composed of chains of nucleotides. But there's only 4 different nucleotides. So, there's only adenine, cytosine, guanine, and thymine. Whereas proteins are also polymers, very long chains of amino acids. Amino acids are the building blocks of proteins, but there are 20 different amino acids, whereas there's only 4 different nucleotides in DNA. There's 20 different amino acids that are contained in proteins. So it just seemed like that protein was the more logical molecule to contain our genetic code. But Frederick Griffith, here's what he did. He injected mice with 2 strains of pneumococcus, which is the Streptococcus pneumonia bacterium. One is the S strain and the other is the R strain. The S strain is virulent, and that means it killed the mice. The R strain is not virulent, and so it did not kill the mice. It didn't make them sick or kill them. So the mice that he injected with the S strain died. The ones he injected with the R strain lived. Okay, so here we have the S strain, which is the blue bacteria, injected into the mice, they die. The R strain, which doesn't have a capsule around it, injected into the mice, they live. So then he heat killed the S strain. He heat killed the S strain. And by heating the bacteria and injecting it into the mice, they lived. Then the next thing he did is he mixed in a test tube live R strain bacteria, which by itself did not kill the mice, with dead heat-killed S strain bacteria. So the DNA from the heat-killed S strain entered into the R strain, and the R strain bacteria that didn't kill the mice now killed the mice. So that was a hint that DNA, that was the transformation. Transformation is when DNA enters live bacteria from dead bacteria. So this is what Frederick Griffith discovered. Then Oswald Avery and other scientists did more work on this, subject of transformation. And what they did is they found that when they added this enzyme called DNase, which is the enzyme that breaks down DNA, whenever they mixed in DNase with the Streptococcus bacteria, that transformation did not happen. So if you go back to this experiment D, which did kill the mice, when you added DNase, which is an enzyme that breaks down DNA, when that was added into the test tube along with the live R strain and the dead S strain, then the DNA from the dead S strain was broken up by the DNase enzyme. It couldn't enter the R strain, and these mice lived. So when you added the DNase, the enzyme that breaks down DNA, then this experiment didn't work. What that tells us is DNA must be what was leaving the dead bacteria and entering the live bacteria. It must be the DNA. So when you combine Griffith's experiments with Avery's experiments, it seemed pretty obvious that DNA is the transforming substance. And then you have Hershey and Chase, and they were working on viruses. They were working on a virus called a T-phage, and it is a bacteriophage. A bacteriophage is a virus that infects only bacteria. So the T-phage is a virus that infects E. coli bacteria. And what they were trying to find out is so the virus basically has 2 main components. it has an inside core of DNA, and then outside it has a protein coat. So it's made of DNA and protein, and that's it. So what they did is they radioactively labeled the DNA, but not the protein, and mixed it all together with bacterial cells and found out that the radioactively labeled DNA entered the bacterial cells. And then when they did the same experiment, but they radioactively labeled the protein instead of the DNA, then there was no radioactive substance inside the bacteria. So by radioactively labeling the DNA and the protein, they could trace what ended up in the bacterial cells, and they knew for sure that DNA ended up inside the bacteria. So it was the DNA that had the genetic code for the viruses, because that's what viruses do. They basically code for new viruses. So it was either the DNA inside the virus or the protein coat that contained the code to make new viruses. And so Hershey and Chase proved that it was DNA. So here is another experiment proving that DNA is the genetic material. And the experiment's kind of outlined here. I'll let you look over that. I really want to move on and talk about DNA structure. So we know DNA is a nucleic acid. It's deoxyribonucleic acid. And nucleic acids are one of our 4 macromolecules. And it's a chain of nucleotides. The building blocks of nucleic acids are nucleotides. So we have 4 nucleotides that are the building blocks of DNA. 2 of them are called purines, and they are adenine and guanine. And from this point on, we'll call them A and G. And then the other 2 nucleotides are called pyrimidines, and those are thymine and cytosine. And from now on, we'll call those T and C. Now, the difference between a purine and a pyrimidine is that a purine has 2 rings. So adenine has 2 rings, guanine has 2 rings, as you can see in the picture, and a pyrimidine only has a single ring. So thymine is a single ring structure, and cytosine is a single ring structure. Okay, so Chargaff is a scientist who discovered that when you analyze the composition of DNA, there's always an equal amount of A and T, and there's always an equal amount of G and C. That suggests that since DNA is a double-stranded structure, that A's always pair up with T's and G's always pair up with C's. We call this complementary base pairing, and this was discovered by Chargaff. Okay? Each human chromosome contains, on average, about 140 million base pairs. So pairs of A's and T's, G's and C's. All right, so on average, about 140 million base pairs. And if you look at humans, there's about the same percentage of A's as T's and the same percentage of G's as C's. And you can see that from this chart. And then it shows you for other species that the same is true. And this is what Chargaff discovered. So A's always pair with T's. G's always pair with C's. Now, the scientists that were given the Nobel Prize for discovering the structure of DNA are Watson and Crick. But they were assisted by the research done by Rosalind Franklin. And Rosalind Franklin studied the structure of DNA by taking X-ray images, X-ray diffraction photographs of DNA molecules. And from her photographs, she found out 2 things. She found out that DNA is a helical structure. It's a double helix, which another way to say that is it's shaped like a twisted ladder, and that some portion of that helix is repeated. Okay, So this is Rosalind Franklin, and this is her X-ray image of DNA. Now, you and I may not be able to look at this shape and tell that that's what a double helix would look like under an X-ray, but